Biomedical importance of glycolysis. Glycolysis is the first step of cellular respiration. It is an important biochemical process which provides energy for the functioning of various organs. The energy is nothing but an ATP molecule, which is adenosine triphosphate. In glycolysis, glucose is converted to pyruvate, and ATP is further produced. Glycolysis occurs in the cytoplasm of the cell. It is very important for the functioning of various cells in the body. The oxygen is carried throughout the body by hemoglobin in red blood cells, then it supplies oxygen to various tissues. The RBCs lack mitochondria, so they primarily rely on glycolysis for energy production. Pasteur effect was discovered by Louis Pasteur while studying fermentation by yeast. He observed that when anaerobic yeast cultures were exposed to air, the utilization of glucose decreased by nearly 7-fold. This shows that the utilization of glucose during anaerobic condition is higher than aerobic condition. Otto Warburg first noticed the difference between metabolism of cancer cells and normal adult tissues. He observed that cancer cells consume glucose at higher rates than normal tissues, but uses a smaller fraction of it for oxidative phosphorylation. Cancer cells also produce lactic acid even when oxygen is present. While normal tissues stop producing lactic acid in these conditions, the amount of lactic acid produced by cancer cells is also much higher than that produced by normal cells.
Rappaport-Lubering cycle occurs in erythrocytes. The oxygen is carried by the RBCs and it is distributed to various tissues.
In RBC, the 1,3-bisphosphoglycerate, which is an intermediate of glycolytic pathway, is converted to 2,3-bisphosphoglycerate by the enzyme bisphosphoglycerate mutase.
Under hypoxic conditions, the 2,3-bisphosphoglycerate concentration increases in RBC. 2,3-bisphosphoglycerate combines with hemoglobin and reduces its affinity towards oxygen. So in the presence of 2,3-bisphosphoglycerate, oxyhemoglobin unloads more oxygen to the tissues, therefore increase in 2,3-bisphosphoglycerate is observed in high-altitude fetal tissues and anemic conditions. In cytoplasm, glucose is converted into glucose 6-phosphate by the enzyme glucokinase or hexokinase.
In hexose kinase deficiency, the 2,3-bisphosphoglycerate level is decreased. This will increase the affinity of hemoglobin for oxygen. This results in the difficulty in releasing oxygen to the tissues. It causes hemolytic anemia and icterus. The symptoms of hexose kinase deficiency are fatigue and pallor. Phosphoenol pyruvate is converted into pyruvate with the help of enzyme pyruvate kinase. It is an important enzyme in glycolytic pathway. Red blood cells do not have mitochondria, so they are dependent on glycolysis. Pyruvate kinase deficiency causes low RBC glycolysis. Low ATP affects sodium-potassium ATPase pump and leads to membrane damage, lysis, and hemolytic anemia. The symptoms of hexose kinase deficiency are fatigue and pallor. During hypoxic conditions, pyruvate is converted to lactate. This lactate gets accumulated, which causes lactic acidosis, especially during high-intense exercise. It results in pulmonary embolism. The risk factors include diabetes mellitus. Thiamine is important for the formation of TPP, which acts as a cofactor for the enzyme pyruvate dehydrogenase. So deficiency of thiamine inhibits pyruvate dehydrogenase activity. In alcoholics, thiamine deficiency interferes in the activity of pyruvate dehydrogenase. Due to this impaired PDH activity, pyruvate gets accumulated and then converted to lactate by LDH, which causes lactic acidosis. Hope you find this video interesting. Thank you.